right. Well, good afternoon, everybody. Hope you've got your lunch either in front of you or in you. Um, Welcome to the fifth and last of our. Um, we've had quite a week, and we have a great one to end with today. Um, these webinars are put on by two organizations, the California Invasive Plant Council. I'm Doug Johnson. I'm the director of CalIPSI, and the University of California's Division of Agriculture and Natural Resources. Sabrina, do you want to give a quick background on UCANR?
Sure. So we're the part of University of California. We're not part of any one campus, but we run the statewide network of cooperative extension offices in every county in California. And we do stuff from agriculture to master gardener programs to nutrition, 4-H. I'm the natural resources advisor in Los Angeles and Ventura counties and do a fair bit of work on invasive species. Um, Please come check us out if you're interested. And I'll just briefly say I'm also joined today providing some technical help by Elliot Frutal, who's with our California Naturalist and California Climate Stewards programs, both excellent programs that are open to any member of the public to take as long as you can find one near you. If you want to get more hands-on involved in things like invasive species management. I also just want to take a moment to give a pitch. You're going to receive a link to a survey a little bit later on. If you've been able to join us all week, you've probably seen that a few times. Um, if you've been at all of them, I really encourage you to fill this out after this one today because it is for all of them. And the results are actually very important to us, both to improve the series as we go through over the years and also to be able to justify spending our time on work like this. So I very much appreciate it. Doug?
Excellent. Thank you, Sabrina. Yeah, we're thrilled to partner with University of California on this effort as well as others. CalIPSI is a nonprofit organization statewide, membership driven, and we have an annual conference. This year is our 30th, and it's online, so it's easy to attend, happening at the end of October. We have lots of resources on our website about invasive plants, so you can find out about all of that at cal-ipc.org. Quick housekeeping. We will use the Q&A on Zoom for questions for the panelists today, and we'll use the chat for, besides chatting, for any technical questions you have. Wanted to briefly give background on the California Invasive Species Awareness Week, which is what this webinar series happens this week in June, is that the state has declared the second week in June as Invasive Species Awareness Week each year. And the state's Department of Fish and Wildlife organizes events across the state and posts a clearinghouse online of events happening in all sorts of local areas. They also have a youth art contest. So feel free to check out that website. As Sabrina mentioned, and I mentioned, this is the fifth of five lunchtime webinars we had this week. All the recordings are being posted online on the Invasive Lunch website through UCANR. And so if you've missed them, a few of the topics we've had are invasive crabs in community science, fire followers, so what weeds come in after fires, and how can community scientists help track those. Lake Tahoe, what sort of community science are they using to track aquatic invasive plants there? And then yesterday, Bea spoke about how community science is helping to protect trees by tracking the presence of pests like shothole borers. And so you may recognize the theme there of community science. And as Sabrina mentioned, the California Naturalist Program is a great way to get involved in that. So yeah, searching for invasive lunch will bring up that website, and you can look for the recordings there if there are any that you missed that you want to hear. All right, well, I would like to end the polling and share the results of the poll. So you should be seeing that Wow, we have a whopping almost 2/3 of folks are from SoCal today, and someone's from another country. 2 people are from another country. I don't know if those watching the chat have seen where that's from, if they've told us, but feel free to shout it out.
Canada, at least one
All
person was
right,
joining us from British Columbia.
excellent. Yeah, Spartina is a problem up and down the West Coast. How familiar are you with the invasive species issue? We have a lot of folks that are very familiar, but we have some that are just checking in for the first time and are not very familiar, which is wonderful. That's something we want to do, is increase awareness out there, and a lot of folks in between. And then favorite bird, well, it looks like a tie between burrowing owl and osprey. And that's it's an interesting pair. um, can anyone tell me if osprey eat burrowing owls? Can't tell me, or they don't? They don't. Okay. Um, and lots of others on here. I'm sure there have been plenty shared in the chat as well. And Sabrina, I think you may have even shared your favorite fish or something.
Yes, and osprey are predominantly fish eaters.
Yeah.
I mean, they're
Okay.
basically
Well,
that's
how
all
about
they
that
eat
northern
unless they're
harrier?
really
Is the northern
hungry.
harrier scoping the burrowing owls at all? Okay. Well, so without further ado, I'm going to close the poll and stop sharing. And I'm thrilled to introduce our speakers today. Calypsy works with the Invasive Spartina Project as of a couple of years ago. The project's been around for a long time and is one of the most impressive, if not the largest and most impressive in California. So I have the honor of introducing Jeanne Hammond and Lindsay Faye Domecus. And Jeanne has worked on the Invasive Spartina project for the consulting firm Olufsen Environmental since 2008, so it's been a good long time. She wears many hats for the ISP, but the main one is as Restoration Program Manager. Before working on tidal marsh restoration projects for OEI, Jeanne worked at Point Blue Conservation Science, conducting songbird research and monitoring projects, primarily in restored riparian habitats. She has a BA in Urban Studies and Environmental Planning from UC San Diego, and an MS in Natural Resources Wildlife Management from Humboldt State. Lindsay Faye Domecus is an environmental biologist at OEI, Olufsen Environmental Inc. She started there as a seasonal employee in 2017 after finishing her graduate degree in environmental science at San Francisco State University, where she studied environmental physiology. And at OEI, Lindsay works on a variety of projects, including the Invasive Spartina Project, and spends most of her time working in the marshes of San Francisco Bay. And both Lindsay and Jeanne agreed that a great work day for them is one that involves a kayak. And they spend lots of time wearing hip waders and getting muddy, which is different from my typical office day. So I'm excited to hear about all the work that you guys are up to.
Thanks, Doug, and thanks everyone for joining Lindsay and I to learn a little bit more about invasive Spartina species in the San Francisco Bay Estuary. And it's not advancing.
Don't you love that?
One moment.
Yeah, you might need to restart it.
Yeah. Hold on, let me get out of here. Sorry, everyone. This was happening earlier to both Lindsay and I, and now I can't even.
That's an amazing photo.
Yeah.
So yeah, in recent years, I've seen from some of the work that one of the partners, Drew Kerr, has done from flying his drone around, some really interesting pictures of marshes from the air, showing, for instance, how it looked, several years back, and then how it's looked after revegetation with native Spartina. And the ability of Spartina to spread is pretty amazing over several seasons. How's it looking now, Jeanne?
I got it. Yay!
All right, take it away.
So first, before I get into the invasive Spartina species, I wanted to talk a little bit about why native tidal salt marshes are important. So when you're flying into San Francisco or Oakland airports, or when you're whizzing across the San Mateo Bridge or the Dumbarton Bridge in San Francisco Bay, you may have noticed, I'm sure you have noticed, the sinuous blue and green of the salt marshes and maybe some of the reds of the former salt ponds. Or maybe just you smelled that funny sulfury smell on a summer day when you lowered the window down. All of those are sort of salt marshy kinds of things, and salt marshes are super important to the health of San Francisco Bay, and also to all of us who live on the edges of the bay. We live and work and play on the bay. So why are salt marshes important? They're highly productive ecosystems that are really rich in biodiversity. They're also important carbon sinks, so they're helpful in combating climate change. They can also help protect coastal areas from natural disasters, acting as a buffer against storm surges and erosion. Essentially, they're the upper vegetated portion of mudflats. So if you think about it, when you're out there looking around the bay and the tide goes out and you see all the mudflat, the upper portion of that is sort of where the tidal marshes are. They're sandwiched between the marine and the terrestrial ecosystems, and sometimes they provide the connection between those saline waters and the fresh waters. So they're super important critical habitat for a bunch of different aquatic and terrestrial organisms that feed and find shelter and breed in salt marshes. Salt marshes also act as high tide refuge for birds that are feeding on the adjacent mudflats. And they also provide really valuable ecosystem services of significant commercial and recreational value to all of us. And one example of that is they really help protect water quality by filtering contaminants from our urban runoff. And globally, we've lost 25 to 50% of our salt marshes. But this graphic kind of shows you in our San Francisco Bay Estuary, we've actually lost more like 85% of our tidal wetlands to human development. And since, so the left-hand side, this is a really great graphic that San Francisco Estuary Institute put together. On the left side, it shows you the historical extent of what we had in tidal marshes. It's about 200,000, estimated about 200,000 acres. And then today on the right side, you can see the really light green is where we have existing tidal marshes, or prior to us starting to do marsh restoration. And the nice thing about this graphic on the right is it really shows the effort that the conservation community around San Francisco Bay has in already starting restoration projects, and in the dark green are some of the ones that are proposed in the future. And the Baywide goal is to get us up by 100,000 acres once all the restoration that's planned here is finished. So thinking about the fact that we've lost 85%, the San Francisco Bay Estuary is still the most expansive area of salt marshes on the West Coast, and it's the second largest estuary in the entire US, so it's still super important. The bay's tidal marshes are extremely productive habitats, and they create a lot of nutrients and a lot of organic material that supports the bay's food web. Marsh plants also they provide protected areas for many animals for foraging and breeding and nesting, including a number of endangered species. It's also really important to have wintering habitat for waterfowl and shorebirds. And of course, we all know San Francisco Bay is a really important stopover area for an estimated more than 1 million birds each year that migrate along the Pacific Flyway. And this slide just shows you, thinking about all that habitat loss, of course, the critters who lived in those habitats have also decreased in number. And these are just a couple of them that we deal with in our project, salt marsh harvest mouse. And these three are all protected by federal and state Endangered Species Act. Salt marsh harvest mouse, California Ridgway's rail, and California black rail. So what are some of the continuing threats to our salt marshes here? And like all salt marshes around the world, the biggest threat is climate change. And, our coastal communities are threatened by sea level rise, and that also applies to our salt marshes who are at the edges of our coastal communities. With that increasing water level, the vegetation is going to get inundated more frequently, and it may or may not be able to persist. It's the effect of drowning of salt marshes is what it's referred to. In addition, there's this shifting precipitation patterns that we're seeing as part of climate change, with less winter snowpack, possibly more winter rainfall, currently extreme drought. I think it's extreme. I don't know if it's extreme yet. It certainly feels extreme. With climate change, we're going to see more frequent and intense storms, which is going to lead to increased erosion. And so how do salt marshes benefit coastal communities? Well, they can mitigate waves. They also protect urban areas from flooding. And I talked about this before, the water quality effects of the marshes that can filter pollutants from our urban runoff. Marshes can also help reduce erosion on adjacent shoreline areas. They stabilize shorelines. Marshes are also known to sequester carbon, so helping with greenhouse gas emissions. And then something that we've all discovered in this past year is that open space for recreation is really critical to our mental health. And around San Francisco Bay, we have the San Francisco Bay Trail, which opens up these areas to us and allows us to get out and really enjoy that open space. So this slide is really just sort of a little tiny window into bay conservation and sort of the little pieces that are relevant to the establishment of the Invasive Spartina Project. I'm sure a number of you are from Northern California, so you may probably familiar or have heard of the Reber plan. With the 85% of the tidal wetlands that we lost, we were going to lose more if this plan had gone into place. Mr. Reber first proposed it in the 40s, and it basically was 2 giant dams that would have gone across roughly where the Richmond-San Rafael Bridge is today, so sort of in the Northern Bay, and then where the Bay Bridge is, which goes over to San Francisco from the East Bay. And behind those giant dams would have been 2 very large freshwater lakes. And in addition, it had called for 20,000 acres of additional fill with a shipping channel and locks leading out to the bay and then the ocean. And luckily, it was completely infeasible, and the Army Corps engineer declared it so in the 60s, but it was around from the 40s to the 60s. And so as part of that, 3 women who lived in the East Bay saw this plan and looked out their windows and thought, oh, gee, my home here in Brooklyn, Oakland, they're going to fill all in front of me. And it basically catalyzed the Bay conservation movement and the women started Save the Bay. And as part of that, they in turn then helped get past the first wetland protection legislation in San Francisco Bay, which included the establishment of the Bay Conservation Development Commission, which is the first coastal zone management agency that was really charged with protecting tidal wetlands and specifically stopping fill of wetlands. And so the kind of vision that the Save the Bay had of changing our bay from being sort of a dumping ground into what we think of it today as this treasured resource, open space and home for critters and everything is really started here. And as we started to think about, we've lost all these wetlands and we want to get them back, the first wetland restoration projects were implemented in the 70s and the 80s. And as part of the overall conservation movement and the sort of importance of coastal resources coming to mind, the Coastal Conservancy was established in the mid-70s and to support projects that protect coastal resources. And while this is all happening in the 1980s, the East Coast species of cordgrass called Spartina alterniflora was introduced in a restoration project by the Army Corps. And kind of, I won't be talking about Spartina alterniflora, I'm going to talk about hybrid Spartina, and I'll tell you how that happened in a minute. But the hybrid Spartina spread from that initial site, and so landowners around the bay started trying to take care of it and realized pretty quickly that a plant that can move around with the tides and the tidal system, one landowner can't deal with it. You need to have a really collaborative, coordinated regional effort, and that's where the Invasive Spartina Project came from. Okay, so who are we and how do we do the work? Like I said, the State Coastal Conservancy is involved as the co-lead on our project with the U.S. Fish and Wildlife Service, and Calypsy and Olafson Environmental manage and do the work on the ground. We also have 200 partners around the bay. You can imagine anywhere that the bay touches lands around the bay is a potential partner for us, because that could potentially be habitat for Invasive Spartina. Those partners include everything from private landowners to cities and counties and mosquito abatement districts and water districts and local, state, and federal agencies. And it's pretty much anybody who has jurisdiction or owns land near the edge of the bay. So first, before I talk about the invasive Spartina, I really wanted to touch on our native Pacific cordgrass, Spartina foliosa. also known as Sporobolus foliaceus, due to a fairly recent taxonomic revision. I think everyone's sticking with Spartina foliosa in the near term anyway, but it's just a fun name to say, so I thought I'd say it. This is a perennial grass in the grass family Poaceae, and it's a rhizonomous grass. So what that means is that it can spread both by seed, but it also has rhizomes, underground root structures that grow laterally and tillers that will come up so that it can spread colonially across a mudflat as well. Distribution of the native cordgrass, it's limited to California and Baja California. And it's really a foundational species in our low marsh here in San Francisco Bay. It's an early colonizer. It can come in via, pieces of the rhizome can actually move around the bay and plant themselves, and a new plant can come up from the rhizome. So it doesn't just spread from seed, it can also spread from rhizomes fragments. It also grows in a really narrow elevational range in tidal marshes, and it kind of stays in its lane essentially. And I included this little diagram. It's a marsh zonation diagram in San Francisco Bay and the right-hand corner of the slide. And you can see, you go in the lower elevations, you're down in kind of bay waters and the subtidal, you come up into the mudflat, and then that next zone up is the low marsh where vegetation can start to come in. And that's where Spartina foliosa, the native cordgrass, grows. And above that, you get into what we think of as the tidal marsh plain, where perennial pickweed grows and other plants, and you have your salt pans and your channels. And then higher elevation, you get into the ecotone, which is sort of that transitional habitat between the tidal habitats and the upland habitats. And the one thing I wanted to say about Spartina foliosa in particular is that I wanted to point out that in San Francisco Bay Estuary, over 99% of the cordgrass that we have is the native cordgrass. All right, so what are the invasive Spartina species that we have in San Francisco Bay? I'm going to talk first a little bit about Spartina densiflora, which is up in the right-hand corner. It's a perennial bunch grass that was originally introduced into the US, into California in Humboldt Bay via the timber trade in ship ballast. And then it was introduced locally in San Francisco Bay as part of a restoration project in Corte Madera Creek watershed, which is in Marin County. It was mistakenly thought to be a form of the native cordgrass. And then in the left-hand side, we have Spartina anglica, which is also the result of that same restoration project. And it's limited to the original introduction site. There's very little of it left. And Spartina anglica, also known as English cordgrass, is actually a hybrid between the East Coast native for US, Spartina alterniflora, and an English species called Spartina marittima, hence the common name English cordgrass. And then in the lower right-hand corner, we have Spartina patens, which is also called salt meadow cordgrass, which is only found at one site in the Benicia State Recreation Area. And these three species I've just talked about are very, very close to eradication. There's extremely small amounts of them left in San Francisco Bay. And our big problem that we still have is the one in the middle, which is our hybrid Spartina. And hybrid Spartina is a hybrid between the East Coast native Spartina alterniflora and our West Coast native Spartina foliosa. I'm going to talk a little bit more about that hybridization now. So how did it happen? Like I said, the East Coast species Spartina alterniflora was introduced here in the 1970s as part of another restoration project by the Army Corps of Engineers. They were looking at ways to stabilize dredge spoils, a bank stabilization project. We know a lot more now about what the possible unintended consequences of introducing a novel species into a different ecosystem. So the hybridization of the native cordgrass with the East Coast cordgrass probably happened sometime in the 80s, and it kind of lurked for a little while and no one knew what was going on. It was eventually discovered in the 90s. And in the meantime, that original hybridization happened, and then there's been a bunch of additional events where there's a lot of back crossing that happened, and which produced a lot of different forms which have some transgressive traits, meaning that they can exploit a lot more of the tidal marsh niches than the original native species. Okay, so this is a little more on the hybrid. On the left-hand side, the illustration is Spartina foliosa, and the other illustration is hybrid Spartina. And then the photo that shows the two different plants, you can see Spartina foliosa on the left and hybrid Spartina on the right. And so something that happens with hybrids quite frequently is hybrid vigor, which means that the hybrid has more robust characters than either parent species. And in this case, the hybrid Spartina tends to have, it tends to be taller, it tends to grow more densely and have greater cover, which means it ends up outcompeting pretty much all other tidal marsh plant species. It also can grow, the inflorescence is longer, clearly you can see in the photo, which means that it can produce a lot more seed. It also, the biggest problem for us really is that it grows in a much wider tidal range than the native foliosa. So I included that marsh zonation diagram here again, and you can see on the left side, Spartina foliosa are native in its really narrow low marsh zone. And on the right side, you can see the hybrid Spartina, which can grow much lower in the tidal frame down into the mudflats, also up across the marsh plain and even up into the ecotone. So it really, hybrid Spartina basically threatens all of those tidal marsh habitat types. So I just talked a little bit about why hybrid Spartina is a problem. You know, it can degrade the existing native tidal marsh ecosystem because it comes in and outcompetes all of the other tidal marsh species. It also changes the hydrology because it's you can see this photo in the top here. It gives an example of this where it's just really tall, dense plants that end up accreting more sediment, and they can grow lower into these channels, and eventually you can see that this channel would just be choked with vegetation, and you end up with a monoculture of hybrid Spartina across the site. Also, because it can grow lower in the tidal frame, it marched out across mudflats, which basically excludes foraging habitat for shorebirds. And there are changes in the invertebrate community as well as a result of that. UC Davis did a bunch of research on the invertebrate community and invaded these hybrid Spartina meadows versus unvegetated mudflat, and found that, you know, the number of invertebrates available in the surface muds decreased by over 70%. And the types of invertebrates that are available also changed. So you can envision shorebirds foraging in the mudflat and how far their bills can go in. You need those surface feeders. And it turns out that when you have a hybrid Spartina meadow, the surface feeders kind of go away and you get these more subsurface feeders. So it also was impacting their food resources. Hybrid Spartina also endangers native Pacific cordgrass. Pollen swamping, much larger inflorescences. If you've got hybrid growing next to the native cordgrass and the pollen goes out there, it can get onto the native cordgrass and end up making the native cordgrass produce hybrid seed, as well as the hybrid outcompetes the native. So eventually, if left unchecked, hybrid Spartina would likely cause the extinction of Pacific cordgrass. I talked a little bit about this in terms of the hydrology, but it also can reduce flood control capacity. As you can imagine in this picture up here, when it grows across the channel, and it's a very dense growing vegetation, that is going to impede flood flows. And so that is possibly going to cause higher risk of flooding upstream. Mosquito abatement districts around San Francisco Bay are actually some of our major partners, because they have found that hybrid Spartina meadows slow down water, create ponding. And when you have ponding, you have more mosquitoes. We have more mosquitoes, you have high risk of diseases like West Nile virus. Also, for all the reasons I listed above, hybrid Spartina also causes failed native tidal marsh restorations, because if hybrid Spartina is present, it's going to be one of the first plants to come in and it's going to outcompete everything else, and you're going to end up with a hybrid Spartina meadow, when what we want from our native tidal marshes is all of that complexity that we see with the channel, the sinuous tidal marsh channels, the mud flats, the ecotone, the marsh plain, the salt pans, that basically can just become a monoculture. Okay, so I threw this slide in here to show how much hybrid Spartina is an ecosystem engineer. So this is showing a site where you can see in that red rectangle just a few hybrid Spartina clones right off the edge of this creek mouth. And in less than 20 years, the amount of sediment accretion and more hybrid Spartina growing, and sediment accretion and more hybrid Spartina going. And you can see that Spartina meadow has moved out significantly into the mudflat. And you can sort of envision what would happen if this had been left unchecked. If we hadn't started controlling for it, it probably would have covered a major portion of our mudflats in San Francisco Bay. So in response to the threats to the estuary, the Invasive Spartina Project was initiated to coordinate estuary-wide Spartina control efforts. And we began full-scale implementation and treatment in 2005. And we currently do annual surveys in potential habitat for hybrid Spartina of over 70,000 acres that informs our treatment program. And this slide just goes into a little bit more detail about the extent. You can see where hybrid Spartina could be distributed around the bay. And from 2005 to 2020, it shows that sort of how things have changed over time. In 2005, well, I should describe the colors. So the dark green indicates places where there's no invasive Spartina, and the dark red indicates places where originally there was more than an acre of hybrid Spartina. And as you can see over time, those red sites go away completely, or not completely, but we're down to very few of them, and we've basically reduced the amount of hybrid Spartina around the bay significantly. And to give that in a more graphical representation, here, we started with roughly 805 net acres in 2005. We're down to 33 acres in 2020, and that's a reduction of 96% estuary-wide. And so we're kind of that critical. We need to keep continuing our project so that we can complete eradication, so to support all of these regional native tidal marsh restoration projects that are coming online. And with that, I'm going to turn it over to Lindsay, who's going to talk about how in the world do we do this?
Awesome. Thank you, Jeannie. Hi, everyone. I'm Lindsay Domecus. I'm one of the biologists that gets boots on the ground for ISP at Olufsen Environmental. And I am going to talk today about our different programs. So we'll start out talking about our inventory program, where we go out and actually ID and map invasive Spartina around the bay. Then we have our treatment program. So we go out and we treat and remove the invasive Spartina that we've found and mapped. We have our Ridgway's rail monitoring program, where we monitor the population of Ridgway's rails throughout the estuary. And we have our revegetation program, which is spearheaded by Jeannie. We add plants into areas that we've removed hybrid and bolster marsh habitat for rails and other species. So I chose this picture because it's a great photo taken from a helicopter by Drew Kerr over Bear Island, which is one of our biggest marshes in San Francisco Bay. And you can just see the vastness of just this small part of Bear Island. And there are two biologists down there. Looks like they're probably doing some inventory. So walking sort of these tight track lines up and down these marshes to look for hybrid Spartina and get it mapped so that we can go out and treat it. So with that, next slide, Jeannie. So OEI biologists inventory up to 70,000 acres of the San Francisco Bay year to year. We don't get to every single one of those acres every year because we have sites that don't need to be looked at as often, but there are years where we get pretty close. So we walk around the bay with handheld GPS units that help us take accurate locations of hybrid Spartina patches. And the benefits of this extensive inventory is that, first of all, it saves time because the treatment crew only has to come out and visit features that have already been mapped. It reduces impacts to sensitive habitat. So biologists are able to lead treatment crews to mapped features and minimize walking through marsh and disturbing sensitive habitat more than is necessary. It also gives us the time to thoroughly inventory the marsh and identify the hybrid Spartina, and that allows us to save as much of the native foliosa as possible. Just to throw it out there, this is not an easy task because Jeannie didn't really go into hybrid Spartina features as in they vary. So usually hybrid Spartina is bigger and denser and more robust, but that's not always true, especially based on where it lives in the bay, also where it is in the marsh itself. And there are just lots of different factors that determine what hybrid Spartina looks like. Time of year matters. So it's definitely an art form and one that we are all perfecting. on a year-to-year basis. And we do an extensive amount of genetic testing every year. So we take approximately 500 genetic samples each year. And we tend to do this in areas where we really want to save the foliosa. So if we have patches of hybrid Spartina that we're not quite sure are hybrid, then we'll do genetic testing, and that helps us inform treatment for the current year. And it also just helps us inform inventory for future years. Next slide, Jeannie. So this is a graphic you've already seen from Jeanne, but I thought I'd spend a little bit more time talking about it and emphasizing the extent of this monitoring that we do widespread throughout the San Francisco estuary. So we have 220 sub areas that represent these 70,000 acres of habitat. And in 2020, OEI mapped 33 net acres of invasive Spartina. That's down from over 800 acres of invasive Spartina 15 years ago. So for context, that 33 acres is 0.0005% of those 70,000 acres. So that's a pretty small number. Things are definitely improving and we're doing a great job, I think. And then Jeanne already broke down what the different colors mean. But basically those greens and yellows are good and the oranges and reds are not as good, but definitely seeing a lot more green and yellow as we're moving forward. And Jeanne, if you'll give the slide a click, very good. So the little channel right adjacent to this star that looks like a little finger kind of coming out and pointing at the star is AFCC, that's the Alameda Flood Control Channel. And that's our original introduction site of Spartina alterniflora, or the beginning of hybrid Spartina that Jeanne discussed earlier. And you'll notice that that channel is green, which means that there's not a lot of hybrid Spartina there anymore. Done a great job of eradicating the issue there. I think there's still a little bit, but we're looking really good there. So yeah, next slide. So then just to dive a little bit deeper into those numbers, as of 2020, 151 of our 220 ISP sites that had historical infestations of hybrid Spartina now contain under 10 meters squared. And the total in that area is 186 meters squared net cover of invasive Spartina. So those are the numbers as they stand now. And just to give you an idea of, I know probably most people here know what a meter squared looks like, but when we go out in the marsh, we identify plants and then we map them, and we have to give a really good estimate of how much hybrid Spartina we just mapped so that we know how much chemical or crew or whatever to bring out so we can do our treatment. So a meter squared for me is my shoulder to my fingertips. So if you just go like this, you get an idea of what a meter squared looks like, and then you can kind of have an idea of what 186 meters squared looks like. So these sites that we work on encompass tens of thousands of acres of marsh and mudflat. So it's a pretty impressive amount of space. Next slide. Okay, so we're going to talk a little bit about how we go out and treat invasive Spartina. So we have a variety of tools in our toolbox, and the reason that we do is because depending on what plant we're treating and what's going on in our treatment program and how we're doing, we can adjust our treatment as needed. So for example, Spartina densiflora, as mentioned earlier in the talk, is a bunch grass, which makes digging really effective for that plant. And then we also use tarps sometimes. So like digging and tarps are the two ways that we can, like tarping, are the two ways that we can eliminate invasive Spartina without using any chemicals or herbicides. And those are pretty effective for certain types of Spartina. And then for our hybrid Spartina, we use a herbicide called imazapyr. And that is because, as Jeanne was discussing earlier, hybrid Spartina is rhizonomous. So if we dug it, we have the potential for missing pieces of the rhizome, and then the plant will come back the next year. So our hybrid Spartina treatment program starts in June. So getting started right about now. And year to year, we treat about 150 sites throughout San Francisco Bay. Next slide. Next slide, Jeanne.
Hold on, I'm trying. It's frozen. Figures, right? Let me. I guess just hitting the button won't really make it do anything. I think I might need to stop sharing to get it to go forward.
Have you tried using the down arrows instead of return?
That's what I'm using.
Sometimes? Okay.
Down arrow, side arrow, any arrow.
If you're on two screens, make sure you click on this screen and then try the down arrow.
Yeah, I haven't moved off of the screen, so.
Weird. Okay.
I know, right? Let me see. Wow. I can't even get out. My screen is frozen. I'm so sorry, Lindsay.
That's okay. No worries. Technology.
I'll use this moment to encourage folks to think about any questions you may have and enter them in the Q&A, and we'll bring those up at the end of the talk.
Jeannie, I also have the PowerPoint, so I have it ready. I can open it if that's helpful.
Yeah, let me see if this works. I'm just going to reopen it like that. I'm going to do this. There, let me see. Oh, yes.
Awesome. Okay, I'm going to power on. So just briefly, we're looking at eradication of Densiflora. So that's the Spartina species that we generally dig instead of using herbicide. Densi sites are historically up in the Marin part of the bay. So we go there twice annually. The first time we go in June when the plant is flowering, and that makes it pretty easy to find. And the second time is in January when the native pickleweed is to nest. So it's brown, so it contrasts really well with the Densi seedlings that are more green that time of year. It's really great to find them in January because once they go to seed, we have a little bit more of a problem because the seed bank for Densi is 5 years long. So if we miss a plant like this one that's already gone to seed, there's a high likelihood that we're going to find a plant there in the next few years. So we try to catch them as many as we can in that January timeframe. So we found 66 seedlings in 2020, and we've reduced Densiflora up in that part of the bay and the bay in general, because that's really only where we find it, by 99.9%. So really great number. Next slide. So imazapyr is the herbicide that we use to remove hybrid Spartina around the majority of the bay. The EPA considers imazapyr practically non-toxic, and concentrations in water after treatment are far lower than any amount that would be of concern for wildlife. And one of the reasons for that is because imazapyr targets plant amino acids specifically. So imazapyr kills plants by preventing the synthesis of certain necessary amino acids that are produced by plants, but not by animals, which is what makes it pretty safe for surrounding wildlife. And this treatment takes place with close monitoring by biologists. So you can see in the photo that I think that's probably my guy Richard out there spraying, and he's got a biologist keeping a close eye on him and has cleared the area beforehand to make sure that there aren't any animals there. And if we do encounter a rail or a salt marsh harvest mouse, we leave the area and get back to that patch another time. So next slide. So the Invasive Spartina Project is a pretty textbook example of a conservation dilemma in a way, because we have endangered species versus invasive species. So human development has led to this massive loss of tidal wetlands in SF Bay, and wildlife that's dependent on those wetlands is now endangered. So human action has then led to the introduction of highly invasive species, which ironically do help create habitat for these creatures that we care about. So how do we balance removing invasive species while protecting endangered species? Well, the answer for us at ISP is adaptive management. So treatment, restoration, surveys to assess population, and then reconsultation for future treatment. Next slide. So here's our California Ridgway's rail, and the photo on the left shows a great example of some hybrid clones that are creeping out and making the marsh bigger. They've created brand new marsh where there was none before, which of course our rails like having habitat, lots of habitat. And then also the hybrid grows tall enough that it provides great cover at high tide. So it's honestly like a great plant for these birds, but it's outcompeted our native Spartina. So what happens is we go out, we're taking out the invasive Spartina, and because it's outcompeted our native is not there to take its place when it's been removed. So removing invasive Spartina creates this deficit in ecosystem function. So that function that's been temporarily provided by hybrid is usually provided by native plants, but they've been outcompeted. Next slide. So how do we keep track of the rail population so we know if our restoration efforts are impacting the species? We conduct call count surveys for Ridgway's rail between the months of January and April at the start of the breeding season. And Ridgway's rails are a secretive marsh bird. So while we see them fairly often because we're out in their habitat, a member of the public is not likely to see one probably ever, unless you're very patient and have some really good binoculars. But if you're out walking in the marshes in like right around dawn or right around dusk and you hear this kek, 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 kek sound, then that's a rail. It's calling not to you, but to one of its fellow rail members trying to communicate something, probably that it wants to mate. But yeah, definitely if you're interested, it's a really distinct call and it's cool to listen for around the marshes of the bay. Next slide. So despite the fact that we are removing habitat, we are seeing rail call counts increase over the last decade. The data here was collected by ISP and our partner organizations that include USFWS and Point Blue. And this increase is due to several factors, which of course includes just bay-wide restoration of tidal marsh wetlands. So we're seeing wetlands where we didn't have them before. We're breaching levees, we're creating new habitat for them, not just OEI, but lots of different organizations and are working on that. And then also just this successful revegetation of places where we've removed Tybrid Spartina, and we're also adding habitat to places we haven't removed Tybrid Spartina, finding great places to add some refuge plants for rails and other species. Next slide. So the star of the show, this Ridgway's rail, is about the size of a chicken. And there he is at the top, and he's really the center of our restoration program. So one of our main challenges is balancing hybrid Spartina removal with the need to protect habitat for these rails. And ISP's restoration program has grown out of that challenge. So that's why we've started our re-veg program. And the main goal of the program is just to rapidly enhance tidal marsh habitat to benefit Ridgway's rails and other tidal marsh dependent wildlife. Next slide. So ISP's re-veg program was started in 2011, and we just completed our 10th year of habitat enhancements. This program is guided by our plan, informed by our tech advisory committee, and implemented at a bay-wide scale. The program focuses on critical components of rail habitat, and we actively use adaptive management to learn from our enhancement efforts. A great example of this is we've learned that caging patches of new Spartina foliosa seedlings helps protect them from grazing from Canadian geese. And we learned where that's important and where that's not based on how our re-veg program did in the beginning and learning where we need to cage and where we don't. Next slide. So this map shows the 40+ sites where we've implemented habitat enhancement throughout SF Bay. The blue areas of the map show planting areas, which is primarily Spartina foliosa and marsh gum plant, Grindelia stricta. And then orange shows where we've added high tide refuge islands. And the photo down there at the bottom shows ISP staff and probably some of our contractors out there getting some marsh gum plant in the ground. Next slide. So here are some great examples throughout the bay of places where we've done some successful revegetation. So the left is, let's see, the top left of this slide here, the two that say 2013 and 2017 are those photos. Thank you, Jeannie. Those photos were taken in Eden Landing, and it was an early restoration project where it was mudflat before, and then we successfully planted Spartina and have this great foliage band going along that channel right there, which is fabulous habitat. And then the photo below that, still on the left-hand side on the bottom, that's AFCC. So that's our original alterniflora and then subsequently hybrid introduction site that we've eradicated most of that hybrid Spartina and planted this great band of foliage that's grown and become just this awesome stretch of habitat along that big major channel. And then on the right-hand side, you'll see two examples. Both of those were taken along the Hayward shoreline of where we've planted marsh gum plant along a couple of channels and just watched it absolutely explode over a couple of years into really phenomenal habitat. Next slide. So just one last example of some great success we've had with revegetation. So this slide shows a photo that was taken in 2011 after this unvegetated mudflat was opened back up. And this site is pretty far from any local source of native cordgrass, which is the plant that we would expect to colonize given the elevation of this mudflat. So we wanted to jumpstart this trajectory of this site to open mudflat from open mudflat to potential habitat for Ridgway's rails. So we did active planting here, which you can see in the next slide. So here are some photo points that have been taken. So we go back to the same location year to year and take the same photo over and over again. And so you can see we have 3 photo points here where there's basically bare mud beginning, and then a few years later, really happy, healthy looking Spartina foliosa. And then on the right-hand side here, we've got a kind of a bird's eye view. This is actually what like our GPS units look like in the field. So those green patches are plantings, probably from multiple years. And then we also have the locations of Ridgway's rails that we've heard during call count surveys. And then that A, B, and C corresponds to those photo points on the left side. So that's where those photos were taken. So we go back to those areas from year to year and see how they're doing. Next slide. And now here's just an overall shot of what that marsh looks like. So you saw 2 slides ago that it was a big open mud flat, and now it's a beautiful, happy marsh. And the best thing is that the rear population at the site is rapidly expanding. We detected 0 birds in 2018, 8 birds in 2020, and 13 in 2021. So within 3 years, we've had 13 Ridgway's rails move in there. Next slide. So sort of to wrap up here, the Invasive Spartina Project is a constantly moving target for us and for all of our partners. And we continue to remove invasive Spartina, restore native marsh habitat, and monitor the California Ridgway's rail. And we hope that working toward this balance can help create and protect healthy native marshes that are more resilient in the face of sea level rise and climate change. Next slide. With that, we'd like to thank our partners. This is just a small spattering of them. We have so many, but we definitely couldn't do any of this great work without all of our partners and collaborators. So thanks so much. And that's it.
Wow. Yes. Sorry, a little slow on the unmute trigger there. That is amazing. Having tried to walk in a mud flat once just to sample some Spartina back in state parks days, I know what a challenging environment that is to work in. So Nikki, what sorts of questions do we have from folks?
Yeah, we have a few. Sarah wants to know, is there anyone monitoring invasive Spartina in the legal delta? If so, do you know where it needs to be managed?
Yes, we're working. We recently added the Sassoon Marsh area as a fairly new monitoring region for us, based on an observation that someone had. Like stepping back a little bit, thinking about freshwater and saline waters. In the farther you go upstream, Spartina species end up dropping out because you get into freshwater species and they tend to compete better. And so you don't really find native cordgrass up the San Francisco or the Sacramento River Delta very far. And so we've looked over the years, but not that much. And recently, I think with the increasing drought years it's probably starting to move up a little more. And so we've started to detect a few patches. There's not very much of it. It's mostly under control. And I'm trying to remember where. So there's a few patches in the Motco area, if you know where Motco is, essentially. I think that's about as far upstream as it goes right now. But we're actively treating them, and they're almost down to zero, I believe, right, Lindsay?
Yeah.
It's a huge area to suddenly have to inventory. We're trying, you know, for the last 3 years, I think we've tried different methods, you know, whalers, it's always windy up there in the Delta, trying to get out to these areas, you know, getting permission to access duck clubs. It's been an interesting undertaking to start getting your handle on Sassoon Marsh area.
All right, great. We had a couple questions about Amazipar from Francis and Naresh. Saying Amazipar is on the California groundwater protection list. Even though the applications are on saltwater marshes, are you folks monitoring its presence in thresholds and adjoining groundwater tables? And along with that, Francis wants to know, what is the half time for Amazipar? Does it, I think, harm any environmental species?
Good questions. I am not the treatment program manager, so I am almost afraid to answer these questions, but okay, say again what if
I understood correctly, it's Imazapyr is an herbicide and it's being applied to the marsh. I know a little bit from hearing from Drew, the treatment manager, that is applied at a certain tide level, and that the half-life in water is quick.
right.
So
Yes.
you want to apply when you have exposure and it's not going to get washed off right away. And then they also do water quality measurements for the NPDES permit all the time, and have found basically none. So that's as far as my knowledge goes, but I don't think of it as a like soaking into soil and getting into a groundwater type situation at all.
Right, yeah, it's approved for aquatic use because it doesn't do that when it's in water. I think Lindsay had a slide on Imazapyr, and I believe there's a 90% reduction in the level in the first week. And what was the other?
Was there a part two to that question, Nikki?
Oh,
yeah,
sorry. Yeah, I thought you meant the slide. Well, we had, are you folks monitoring its thresholds and joining groundwater tables? You touched on that, Doug, with the permit. It doesn't harm any environmental species.
The NPD, as part of the water quality monitoring that we've done every year for the NPDES permit, it's not even persisting in the water. So I don't think you would be able to detect it in the soils at all. Remember, this is a tidal system, so anywhere where we're using it actually becomes inundated twice daily by water, by tidal waters. And and the way Drew has to plan the treatment is predicated on that. He basically comes in and says, okay, the tide, he needs to treat on an outgoing tide so that he has the maximum amount of time of dry time for the herbicide. Because once the water comes back in, it wipes it right off the plants and makes it, it has a certain limited time that it can act on the plant and get absorbed and drawn down to the roots, because we're in a tidal system. And none of our Spartina isn't places where there isn't tidal flushing, essentially.
And I can't remember if it was Elizabeth Versati's research or somebody else's on the benthic invertebrates in the mud under the Spartina. And I don't remember also if that was looking at the effects of the hybrid versus the native, or if it was looking at herbicide impacts. But I feel like a lot of this has been really well studied. And I imagine that there are resources on your website looking at that. Other questions, Nikki?
Yeah, we had one from Eva. How might suggested thin lifts raising the surface of the marsh, anticipating SLR, affect the various species of Spartina and annual marsh species?
So thinking about climate change and how sea level rise will affect salt marshes in general, it's an unknown. I think people, the worry is that with sea level rise, salt marshes are not going to be able to keep up with the pace of the water levels rising. And something we know about San Francisco Bay, and probably a lot of places, is that we're also moving into an area where we have a sediment deficit in some parts of the bay. And so there really are questions about needing more sediment. You know, a lot of our sediment that traditionally was coming into the bay is now trapped behind dams. And thinking about, and there's a lot of people working on this, you know, also part of the Bay Conservation Development, BCDC, you know, is to prevent bay fill. Well, now we're moving into an era where we want to do beneficial fill to help augment the salt marshes that we do have. And a lot of the tidal marsh restorations that are happening around San Francisco Bay are taking that into account and trying to find sources of clean sediment and build up that elevation before they become tidal again, so that they can keep pace better. Because the best thing we can do is to have these tidal marsh restoration projects as far along the trajectory as possible to native marshes or mature native marshes, where they have a lot of vegetation that can help trap what sediment is in the water column and help them keep pace with sea level rise.
Thanks, Jeannie. Real quick, Nikki, I just want to point out to folks, I realize it's a little after 1 o'clock. If you have to go, thanks for joining us, but we'll hang around and answer questions for as long as there's interest. Claire did put the link to the feedback form in the chat again.
And I just really want to encourage everyone to fill that out. We really use that information both to improve our program every year and to justify the time and effort the ANR and CalIPC team are putting into these workshops. So please fill it out.
Thanks, Sabrina. So Nikki, other questions?
Yeah, we have one more question, unless people want to add some more in there, from Bree. Is your district CCC office pushing you away from herbicide use? We've had great success using it in Santa Barbara County for Rundo, but are losing the ability to apply it in the coastal zone.
Not so far. We've been very lucky. Yeah, no,
do you know the CCC reference? Do you know what that, are they talking about Coastal Conservancy or Conservation Corps or Ag Commissioners or?
Well, in general, I know
Coastal
that
Commission.
The Coastal Commission.
Yeah, that comment just came in.
Not so far. I have not heard from our treatment program manager that we've had any pushback about using herbicide in any area that we work in San Francisco Bay. There is, you know, there are, we try to use, you know, a judicious amount of herbicide that's needed as appropriate to do the control of Spartina. There are areas of San Francisco Bay that are very sensitive to herbicide use. And so in particular with like the Spartina densiflora that we talked about, the perennial cordgrass, bunchgrass that is in Marin County, the method that we have used there, which also happens to be the best method for eradicating that species, has been primarily digging, which has helped us a lot with that kind of anti-herbicide general sentiment in the area.
So, and when you mention parts of San Francisco Bay are sensitive to herbicide, you don't mean the habitat is sensitive, you mean the human, the residents, yes, the folks are concerned about herbicide use in general.
Right.
All right, and then we had another question from Matthew. Is aerial or satellite imagery useful in detection or monitoring of different Spartina species populations?
We really wish it would be, and I think the technology is slowly but surely getting there. Unfortunately, because the signatures that you get from cordgrass, whether it's hybrid or native, are very similar, it's really difficult to distinguish between them. So you can see in satellite imagery and or remote sensing data, you can see differences in, say, a monocot versus a dicot, or, you know, pickleweed versus cordgrass, but within the cordgrasses, no, it's not helping us much. We're trying to, like, looking at using drones to be able to monitor areas from above to see if we can detect a patch that way. But so far, those great technologies are just not really available to us on this project.
Okay, thanks. And that's all the questions we have.
Thanks, Nikki. Well, and so if I understand Jeannie and Lindsay, the phenology is different. And I mean, it varies enough that you can't just say, oh, that one's 2 times as tall as that one. That's obviously the hybrid. That's obviously the native. Can you just mention the genetics testing that you do to confirm what any particular Spartina, what the genetics are?
We have a geneticist on staff, and he basically, our genetics were developed by UC Davis researchers out of 2 different labs at UC Davis, and we use microsatellites to look at 8 different genetic markers to determine whether or not it's hybrid Spartina versus Spartina foliosa. And then our geneticist, there was a lab down in LA that we used that used to be part of UCLA's Human Genome Lab, and they process our samples for us. And then our geneticist puts the results of the PCR analysis through the program structure to determine, and that's how we figure out whether it's the native versus the hybrid.
All right, and Matthew also added in, he wants to know, has there been any use of infrared or multispectral cameras?
Not so far.
For the remote sensing, and I think even LiDAR was part of it to look at the stature of the plant. I mean,
Yes.
my impression is that everything's been tried so far, and the multispectral signature, like you say, is it's difficult to distinguish between different varieties of Spartina.
It also, I was just going to say, it also depends, like you were saying earlier, on the time of year. You have to be really careful about, and different sites grow differently and have different inputs. So in the extreme South Bay, the native cordgrass grows much taller than it does in the Central Bay or the North Bay, because there's a lot of freshwater input from sewage treatment plants upstream. So you have to know where you are in the bay, and a lot that goes into it.
A lot of variables. Well, and I know there is a fair amount of remote sensing going on for the South Bay salt ponds restoration, and that looking at Spartina and some other invasive plants is part of that. So hopefully we'll be doing as much as the technology will allow us to do, because that's more cost effective and perhaps less invasive as well.
We had one last question, which someone was asking, Ava Buxton was asking a follow-up on, which is associated with the sea level rise question, the thin lifts, specifically how they are affecting the other Spartina species and annual species.
I don't know much about that. We haven't been involved in those, the projects that are doing that. A lot of them are in the proposal stage, I think, where they're hoping to do it. A lot of the sediment additions that have actually occurred were prior to actual tidal restoration. So they've gone in and taken the former salt pond before they breached it and added in sediment to bring it up to more close approximation to what would be a tidal marsh elevation. The actual sediment additions is sort of, everyone's hoping we'll get there, but I don't know of any big projects that have done it up here. I know that you've done it in Southern California. Are you from Southern California and that's what you're referring to? And like, I don't remember.
We're not sure where Ava's.
Okay.
Thanks, Jeannie.
I wanted to make a comment from way back in the very beginning of your talk, just as the resident fish head. One thing to add to your slide about the benefits of coastal salt marshes is their importance as a nursery for fish and other aquatic organisms, including, if you're sort of trying to couch in ecosystem services, including a lot of species that we, actively use as a fish resource.
Absolutely.
So another great reason for all this work you're doing.
Good point. Thank you, Sabrina. Well, thank you very much, Jeannie and Lindsay, for your time today and your presentation on this amazing project. I think there's a lot we can learn from it. This was recorded and will be posted online. So if you want to look at it again or point somebody else to it, you can. Thank you very much for joining us today. And Sabrina, any last words from UC ANR?
Just in addition to thanking our fabulous presenters, Jeannie and Lindsay today, and everyone who spent their time with us this week. I just want to thank all of you who've hung on to the end here. Thank you for coming, and we look forward to doing this next year.
Thanks, Sabrina. And yeah, once again, if you can fill out the survey, very helpful. All right, have a great rest of your week, and keep invasive species in mind all year long until our next invasive lunch series next year. Take care.